Welcome to my Unit 3, Principles of Physiology, Essay Question 2 video. So let's jump right in. So we got our pressure volume loop here for one cardiac cycle. So our pressure volume loop is going to let's start out at ventricular filling in diastole. So we have the rapid inflow phase of diastole, where we're going to see a steady increase in volume, a large increase in volume, but we're going to have a little increase in pressure. It's not going to be very much. And then once the pressure of the ventricle, we're talking left ventricle here, once the pressure of the left ventricle gets higher than the left atria, our mitral valve is going to close. But our aortic valve is still not open yet because the ventricle pressure is not higher than the aortic pressure yet. So while the ventricle is contracting for this short moment, we have what we call isovolumeric contraction or isovolumic contraction. And so what's happening here is we're seeing a very steady rise in pressure with zero change in volume. Once the ventricular pressure exceeds the pressure of the aortic semilunar valve is going to open up and we're going to have ejection. And then during ejection, the pressure is going to rise and then fall below the level of the aorta, and then we get the aortic valve closing. This is where we hear our second heart sound. We hear our first at the mitral valve. And we're going to get our dicrotic notch here as the elastic aorta rebounds and then causes blood to reverberate onto the valve. And we get that dicrotic notch. And then we're going to have isovolumeric relaxation, which is a steady decrease in pressure, but there's no change in volume at all. And this is because the aortic valve is closed, but the mitral valve is not open yet because the ventricular pressure is still higher than the atria. So we have to relax that ventricle until the pressure is below the atria. And then that mitral valve is going to open back up, and then we restart with ventricular filling. So a couple terms here to go over. We have inotropy. This is our force of contraction. We have afterload, which is the pressure that the heart works against. Some things that can increase this would be viscosity. We could have vasoconstriction. We could have anatomical defects in the valves, things like that make it harder to get blood out of the heart. And then preload is going to be how much our ventricles are filling with blood. And we can think of this as like stretching the ventricle. Like a greater stretch of the ventricle gives us greater preload, which is going to allow us to pump more blood out of the heart, since those sarcomeres are in an ideal position to create force when they're a little bit more stretched out. So some things that can affect inotropy, afterload, and preload. So sympathetic, parasympathetic stimulation are going to affect our inotropy, which is our force of contraction, right? And what that would look like, inotropy doesn't really necessarily affect pressure or volume, but we could just draw like a dotted line that goes up there, which represents our inotropy. Inotropy is like the change of pressure over the change of volume, right? So we can think of this as if we shrunk down to a ventricle wall right at this point when the ventricle is maximally contracted. Inotropy is like the amount of pressure we'd have to put on the ventricular wall to get it to move and create volume. So greater inotropy is like it takes more pressure, and that's more elastance, is we'd have to use more pressure to get the ventricle wall to move. And then compliance is change in volume over change in pressure. So that's like that's like how easily the vessel dilates. That's like how easily you could stretch that vessel in response to change in volume. And then So if we had preload, our graph here would, if we had increased preload, our graph here would get wider because we're filling with more blood. If we increase afterload, then our graph's going to get taller because we're producing more pressure in afterload. And yeah, so that's kind of the pressure volume loop there. So then we have this thing called mean arterial pressure, which is like our blood pressure. It's like our average blood pressure throughout our entire circulatory system. And the equation for this is going to be our cardiac output times our peripheral resistance. Cardiac output is heart rate times stroke volume. Peripheral resistance, the equation for this is going to be 8 times the viscosity value times the length times pi and then radius of the blood vessel to the 4. Because if we have increased, so let's say we have increased viscosity, how would that affect our mean arterial pressure? Well, increased viscosity is going to increase afterload because the heart has to overcome that inertia of that heavier and harder to move blood. So that's going to increase our peripheral resistance if we increase our viscosity. Same thing with our length. The longer a tube is, the harder it is to, say I had a really long straw and I was trying to blow water down. If I was trying to blow water all the way down a 10-foot straw, I'd have to put a lot more pressure in to do that. Whereas if I just had a normal restaurant straw, it takes a lot less pressure to just blow some water through that straw that's shorter. So our increased length increases peripheral resistance. And then we have the radius of the blood vessel. So when we dilate blood vessels during vasodilation, our peripheral resistance is going to go down. And notice that the values on top are being divided by this radius of the blood vessel, right? So if we have the greater the diameter of the blood vessel, the greater the radius, the less peripheral resistance that we're going to have. And then our flow rate, so this is the rate that blood flows through the vessels, is going to be pi times the change in pressure times the radius over 8 times viscosity times the length here. So our values are basically just inverted here, right? So how would length affect the flow rate? Well, if you have a longer blood vessel, you're going to decrease the flow rate through it. If you have now the reason that vasodilation really increases the flow rate is because we can see that here with this equation. If you increase the radius of the blood vessel, that flow rate's going to be higher, which means that blood is going to be flowing faster. And then if you increase the viscosity higher, we're dividing that flow rate by our new viscosity that's higher, so we're going to have a lower flow rate. And then for this class, flow rate is just going to be P1 minus P2 over PR. So if you have a tube and at the arterial side you have a pressure of 50, and then on the venous side you have a pressure that's like 10, then you would subtract 50 minus 10 and get 40, and then divide that by your peripheral resistance. So let's say that value is like 8, then you'd have like a flow rate of 5. So this is just a simplified version of the equation above. But basically what it's saying is that if you have really high pressure on one end of a tube and low pressure on the other, that flow rate is going to be, if you have high pressure on one side and low pressure on the other, combined with low peripheral resistance, you're going to have a faster flow rate. But if you have, high peripheral resistance, your flow rate is going to be slower because it's harder to move that fluid through that constricted vessel there. All right, so now let's move on to baroreceptors and how these work. So a good example of how baroreceptors work is when you're laying down and then you stand up. So you have a change in blood pressure, and that's called orthostatic blood pressure, which is the blood pressure based on the body position. So if you lay down and then you stand up, we get a decrease in blood pressure because there's a decrease in venous blood to the heart because that venous blood is pooling down in our legs, right? And so what happens is we have these baroreceptors on the arch of the aorta and in the carotid sinuses, which are going to sense this decreased blood pressure. And these are stretch receptors. So these baroreceptors are going to sense this low blood pressure as low stretch. And then they're going to send less action potentials to the medulla oblongata. And then what's going to happen is the medulla is going to stimulate the adrenal cortex of the kidney. We're going to release epi into the bloodstream, which is going to increase our heart rate, which increases our blood pressure, right? Because if we come back to this equation here, mean arterial pressure is cardiac output times peripheral resistance. So if we go ahead and increase our heart rate, then our cardiac output goes up. And as long as our peripheral resistance stays the same, or maybe even goes up, we're going to increase that mean arterial pressure, which is going to increase our blood pressure. And the reason that we have this is because if your mean arterial pressure drops below your intracranial pressure, due to this equation here, right? If P2 is higher than P1, then you're going to have a backwards flow rate. It's going to be negative, right? So say P1 is our low systemic blood pressure. Let's say it's 50. And then our P2 is intracranial pressure. Let's say that's 55. P1 minus P2, it's going to be negative 55. And then we divide that by our peripheral resistance. So if systemic blood pressure is lower than our intracranial pressure, then we're going to have a net backwards movement. So that means we're not going to be perfusing the brain. We're going to go unconscious and we're going to die. So this is why we have baroreceptors, is because we need to boost that blood pressure so that we can perfuse the brain. And then these receptors also sense increased blood pressure as increased stretch, which is going to increase the number of action potentials sent to the brain. And then the medulla is going to, via the vagus nerve, go ahead and lower our heart rate. And once again, if we lower that heart rate, then we're going to lower our cardiac output, which is going to lower our blood pressure so that we don't stroke out. So that's a little overview of the baroreceptors right there. All right, let's go over capillary exchange next. So the capillaries and capillary exchange is the whole reason why we have the circulatory system to begin with, right? The point of capillaries is to perfuse the tissues on the cellular level, keep those cells alive. So the capillary is about 1 red blood cell thick. And what happens is the nutrients, the oxygen that the cells need are going to diffuse from the capillaries onto those tissue cells. That's the idea. I just got asked on a group date. I just looked down at my watch, so that's pretty cool. Anyways, so let me put it this way. So now the question that we need to ask ourselves is, I guess, what can go wrong here? Because this is physiology, right? So in order to determine what can go wrong, we need to kind of determine the normal state of our capillary. So this is our arterial side of the capillary, and it's controlled by a capillary sphincter, which is just some smooth muscle that opens in response to a buildup of the products of cellular metabolism so that we can remove that waste and then also get more nutrients in. They're regulated by autoregulation. Those capillary sphincters are not regulated by the nervous system. So say these are open, we get blood flow in. Well, the blood pressure, so we have a factor here called hydrostatic pressure, which is influenced by our blood pressure. We just call it hydrostatic pressure in the capillary. By the time that arterial blood gets from the heart to the capillary, we've gone from about 120 millimeters of mercury systolic pressure to about 30. And that is a force that is causing fluid to leave the capillary there. But within the capillary and within the blood, we have what's called colloids, which are proteins that don't diffuse across the membrane, but they stay kind of afloat in the blood. They're too light for gravity to affect them. And the main protein in the blood that does this is albumin. It's produced by the liver. So albumin is going to be very important osmotically for capillary exchange, because the high concentration of albumin in the blood is going to draw fluid from the interstitium into the capillary. And we have a force of about 28 millimeters of mercury that is going to be involved in this. But then we have our interstitial colloid pressure, which is water leaving the capillary and going into the interstitium, and that's about 8, because there's proteins and colloids in the interstitium as well, so some water has to move there too, right? And then we have an interstitial fluid pressure of, it's about 3 going out, but we call it kind of -3, and that's caused by these lymph vessels. So what happens here is we have more, if we look at our venous compared to arterial net filtration rate, we have a greater osmotic force of fluid leaving the capillary. And if that's the case, there's more fluid leaving the capillary than returning, then we need a method to suck up some of that extra fluid so that we don't start to swell up and have too much edema. And so we have the lymph vessels for that. So those lymph vessels are going to suck up all the fluid that seeps out of these capillaries that is unnecessary. And that's a force of about 3. It's like a vacuum. And then you have your venous. So throughout the capillary, the pressure is going to drop from 30 on the arterial side to about 10 going out on the venous side here. And then, so this results in an arterial net filtration rate of 13 millimeters of mercury going out, which means that on this side of the capillary, we're going to be sending fluid and nutrients out of the capillary. On the venous side, we have an NFR of 7 in. So we're going to have more fluid coming in to the venous and the venule. So basically, some things that can mess this up. So if we take, for example, let's do a chemical burn to the skin. So let's say someone dumps lye on your skin. That's going to kill the skin cells. That's going to kill the cells of the tissues of your arm. And those cells are going to lyse, and they're going to spill their contents into the interstitium. Those contents might be ribosomes, channels, organelles, just all the proteins inside the cell. And those proteins are going to be osmotically active. So that increase in ICOP is going to cause more fluid to leave the capillary and go into the interstitium. And this is going to cause edema because our lymph node is only our lymph vessels are only calibrated for normal ranges. They're not going to be very successful at sucking up this extra fluid that is caused by osmosis the water following those new osmotically active particles. Our lymph vessels aren't going to be able to deal with that. So we're going to have an abnormal collection of fluid in the tissues. They're going to swell up, and that is edema there. Another example of edema would be left-sided heart failure. So the right side of the heart pumps blood into the lungs. The pulmonary vein brings blood into the left atrium of the heart, which then dumps blood into the left ventricle where it's pumped to the rest of the body. But if we have left-sided heart failure, say we've had a heart attack and we're not pumping blood effectively anymore out of that left ventricle, we're going to have a backup of blood into the lungs here. So what that's going to do is it's going to increase the pressure. It's going to increase the hydrostatic pressure on the venous end, which is going to cause more fluid to leak out into the alveoli of the lungs there. And we're going to have pulmonary edema from that. Another example could be starvation or dilution of blood plasma. So if we have less albumin, then what's going to happen is we're not going to be as osmotically active for fluid to come into the capillary, right? So if we have less albumin, our BCOP is going to be lower, which means that we're going to leave more fluid out in the interstitium because that water doesn't want to come into the capillary as much because there's not as much albumin. So if we have starvation, diluted blood plasma, there's less protein in that blood, then there's going to be more fluid outside of the blood in the interstitium, causing edema. So those are a couple examples, and thanks for watching.